Welcome to video 2.4. In this video, I'll be covering the polymerization of nucleotides, those monophosphate nucleotides that make those long DNA and RNA polymers, and we'll talk about the directionality of DNA, which is how DNA can actually be read. First, let's talk about the formation of nucleotide polymers, as the title so nicely says. So chemically, we can react phosphates with alcohols. So from a pure OChem perspective, here we have a phosphate, right? A phosphor connected to a bunch of oxygens, connected to some sort of R group, R1, I don't care what it is. Here we have an alcohol, some sort of OH group connected to R2. Now, without getting into the mechanism, which you don't really need to know, effectively these two molecules will combine and spit out a molecule of water. Importantly, and maybe I'll keep the colors correct, those hydrogens will be these hydrogens here, and you'll actually see the blue oxygen connected to the phosphate is going to be kicked out. So two hydrogens and an oxygen come together, leave as water, right? And because it leaves as water, we call this a dehydration reaction, right? We're losing water. And the consequence is that phosphor is now directly connected to the alcohol. And you can see that the R1 group and the R2 group, right? These were previously different molecules. They were not connected to each other. They have now been connected to each other. And you'll notice there's a new bond that's formed, right? So this bond right here, and I'll actually kind of cover the whole thing, right? On one side, we see that there's a phosphor, a double bond oxygen, NO and an R. That's a phosphoester bond, right? But we also see a phosphoester bond on this side. So we call this, right, the resulting bond, I'll even write this out. The resulting bond, when you combine a phosphate with an alcohol, is a phosphodiester bond. And so you've probably seen this word, and now hopefully you can understand what it means. There's one phosphate that's participating in two different phosphoester bonds at the same time. We call it a phosphodiester. This is the backbone of DNA, truly. Of DNA and RNA, of nucleic acid polymers. So that's the chemical approach. Let's see it more in DNA structure. So DNA formation. Here we have our sugar connected to guanine. I'm not going to draw the full DNA bases to make things easier. We know it's DNA because there's no 2' OH, and we have this C as well. Now what happens is we have the alcohol from this sugar, the phosphate from this sugar. They're going to combine, right? And what we're going to end up with is a water kicked out, and this top molecule G connected by a phosphate. So I should say this is going to go with one of these hydrogens, if there's a hydrogen here. So those two things will go. Where's my orange? There it is. These two things will go, and then the oxygen will be connected to the phosphor there. Right, so now we have a sugar, a phosphate, a sugar, a phosphate, and you can imagine we could have another sugar, another phosphate, another sugar, another phosphate. And if you do that enough times, you have the entire human genome, right? The instructions that encode what is a human being, from a biological standpoint anyway. So you can see that we have a G and we have a C, and now we have a sequence of different kind of letters of the DNA alphabet, which can encode information. That is how polymerization of these monophosphates works. Now, in order to read this, right, we have to understand this concept of what's called directionality. Really crucial stuff, because if DNA is not read correctly, then the human does not work. And to understand directionality, let's just focus on language first, right? At least for English, right? Different languages have different directionality. We read from left to right. That seems kind of silly to say, but if I read this from right to left, I would see E, G, N, A, U, G, N, A, L, right? That's the reverse of language. But suddenly, this no longer makes any sense, right? We lose all the meaning. All the letters are still there, right, in a different order, but since we read it in the wrong direction, we've lost all meaning, right? We don't want to do that with DNA. So how does the body, how does DNA make sure the information is read in the correct direction? For DNA, now on paper, we also read it right to left, left to right, which is kind of neither here nor there, but we assign what's called directionality. We say there is a 5 to 3 directionality of DNA. What that means is that on the left side, for English speakers, right, we have the 5' end. That's the start, I should say, right? The end of the DNA is on this side, but it's the start part. And at the end, on the right side, we say this is the 3' end. This is the end. So GCCATA encodes the meaning. But if I was to read this backwards, ATA, or sorry, I'm just copying it. To read it backwards would be ATA CCG, right? If this is the 5' end, then this is correct. These are the same. But if I assume that the 5' end is over here, this is now a different DNA sequence, right? It means ATA CCG is different than GCC ATA, right? So the directionality is super important, and we're going to understand where this comes from the actual structure of DNA that we talked about. To do that, and understand the 5' and 3' ends, we need to understand the direction of DNA by looking at the numbering of the ribose carbon. I brought this up briefly before, but remember that the sugar is connected, so the DNA base G, C, A, or T in DNA is connected to this carbon, carbon 1. If you're confused about how you can know it's this carbon, since all the other carbons look similar, keep in mind that this carbon is the only one that has 2 bonds directly to oxygen. Right, so here we see that carbon 1 is connected to this oxygen of the ring, and it's connected to this oxygen. That's how you know. Or it's connected to the DNA base, right? Whereas every other carbon is only connected to one oxygen, or one oxygen, right? So that's your hint, and then you go around in a circle, right? The one that's sticking out, that's not in the ring, that's carbon 5 always. So when we say 5 to 3 directionality, we're actually referring to carbon 5 and carbon 3 here, right? If it's DNA, by the way, this OH wouldn't be here. So when you say 5 to 3, we're kind of talking about this direction, 5 going to 3. So what does that mean? Let's look at a DNA polymer to understand it. So DNA polymer directionality. Here is a DNA polymer, and we can see that we have a C for cytosine, an A for adenine, and a T for thymine, right? That's DNA. And again, we read this DNA 5' to 3'. So what does that mean? Let's take a look at the first sugar of the C, the cytosine. So we see here that we have carbon 1, 2, 3, 4, and 5. And you'll notice that on the 5' end, right, the 5', that's where the phosphate is. It goes on carbon 5. So on the 5' end, we have a free phosphate. And free is really an important word here, right? It's totally by itself. If we connected another sugar to it, then it wouldn't be the end anymore. It would just be like this phosphate or like this phosphate, right? It's in the middle. But the end of the DNA chain, there must be a free phosphate group, right? That is the 5' end of the molecule because that free phosphate is on the 5' carbon. So what we assign is the side of the DNA that has the phosphate, this is the start, right? So we're going to read this CAT, not TAC. We don't end here on the 5' position with the free 5' phosphate. We start there. Right? As we go down, we can see here, right, the 3' phosphate, the 3' sugar, the OH of the sugar, is part of that, right? That bond that we talked about. Remember what we called that bond, right? Phosphodiester. It's involved in 2 different ester linkages. And so all of these are phosphodiester bonds, right? Until we get to the end of the molecule. Here, if we number the thymine sugars 1, 2, 3, 4, 5, carbon 3, right? And this is going to be important here. Carbon 3 is now the end of the molecule. Right? At the end on this side, we have a free OH group on carbon 3. So on the 3' end, there is a free 3' hydroxyl group, or OH, on, and we call it 3' because it's on the 3rd carbon. right? So this is going to be the end of the molecule, and this is the start, right? Now, a molecule is a molecule. The whole thing is one chemical, right? Keep that in mind. But when we talk about directionality, we're talking about how am I supposed to read this information? If I read it correctly, from 5' to 3', from free phosphate to free hydroxyl, it reads as cat, right? That has a nice meaning. If I read it backwards, it reads as tech, which I don't know what that is, right? We lose information. So it's important to understand that the directionality of DNA, it actually comes from the ends of the DNA, right? And with that in mind, that's the concept of directionality. So as a last practice problem, right? Since we've been talking about the structure of DNA and RNA, pause the video here and try to convert the following DNA sequence into the actual chemical RNA sequence. Draw it on paper and see if you can make the corresponding RNA sequence to this DNA sequence, right? But I don't mean make it a total, like it's not going to be binding or anything, but change the structure to make it look like RNA, right? So to understand this, I will go ahead and duplicate this just to save some time. And the first thing that we'd have to notice is that all of these OHs, these 2' carbons, need their hydroxyl groups. So by putting all those hydroxyl groups there, we are now much closer to RNA, right? That's the first thing. The second thing is that anytime we see a T, right, that can't be present in RNA. Thymine is only present in RNA, or in DNA, right? These two are fine, but thymine has to turn into uracil. So reading this from a 5 to 3 direction gives us cat. Reading this from a 5 to 3 direction gives us cow, but spelt this way. Right? So with the addition of the 2' OH and changing the T for a U, we've made the RNA equivalent to that DNA molecule. Hopefully you found that question somewhat interesting. And without further ado, we'll go to the importance of that OH in stability and talking about DNA versus RNA, their function and their stability in the next video.